Most welcome my friend in our channel. We are going to understand the basic mechanism of actions antibiotics. When you are going to understand actions of these cell wall synthesis, there is cell wall on which vancomycin, bacitracins, all the beta lactams antibiotics action in their cell walls. While the cell membranes polymyxins are attacking on these regions. When you are going to understand nucleic acid synthesis, where the molecules attack, DNA gyrase, quinolones, RNA polymerase, rifampicins, 50S subunit of RNA molecules and 30S RNA molecules, tetracycline aminoglycosides, and these are the antibiotics actions. Folate biosynthesis also attacked by some molecules. So we need to understand these antibiotics in the details in later section of this video. Let's start. Basic mechanism behind the antibiotics mode of actions. Bacteria sometimes resistance to these antibiotics, but usually these antibiotics kill or inhibit bacteria by inactivating and neutralizing essential cell proteins inside the bacteria outside the cell wall. When bacteria become resistant to these antibiotics, this essential proteins is either removed, eventually this changing essential protein of bacteria discourage antibiotic attachment with that cell. Actually the mean is that when the antibiotic attack on a specific target site, but the bacteria try to remove it by making some mechanism where this antibiotic is not going attack on that particular site. They are hiding. They can make some type of mechanism where the antibiotics is not capable to make action on them, so they can resistant against these type of bacteria. Like most of the genetic information in this universe, in this planet, in this whole system, just like animals and plants, these are changing. They are going to mutation day by day, per second, per minutes. Just like that, in bacterial genomic structure also changing spontaneously, either by the mutation regions. The result of this small changes that can make a large number of the constitutions of the genetic structure changes. So when these type of the antibiotic attack, there is no binding site previously that were presented, but second generation here, upcoming generation of the bacteria, they have no such type of the binding site. So the bacteria are going to resistant in this type of antibiotic because antibiotics are not capable to bind that specific site of that region. So they cannot destroy, they cannot make dysfunction of that proteins. This type of the neutralization is specific protein. Most of such type of the genetic restrictions are presented after the mutational changes. Yes, random changing, spontaneous mutation, that crucial reason behind the antibiotic resistant systems. Many scientists believe these random changes produce large number of target enzyme antibiotics. Researchers are assuming to produce so many target sites of an enzymes, any type of antibiotic, they can be against work such type of the bacterial infections. So moreover, in coming generations, we will see what the research suggests so we can overcome resistant mechanism of the bacterial systems. Because many enzymes have dynamic process, dynamic specific sites where they can attack on them, because mutation changes are going day by day more as compared to them. So these drugs can permeability more functions and they can attack on them. Hope you like my video. If you really like, please subscribe my channel. Thank you so much.